Nicholas, good evening. We're so happy to have you here.
Good evening. Thanks for inviting me.
Not a problem. You have been a author of several books: Before the Dawn, The Faith Instinct, A Troublesome Inheritance, and you have been science editor at New York Times for 6 years, from 1990 to 1996. How did you get started on this career, and what are your interests?
Well, my first job out of college was almost my first job was working for Nature in London on their news staff. And then I moved over to Science, our American competitor in Washington, where I worked for 10 years. And then I left Science to join the New York Times as an editorial writer. So I wrote about, political issues like environment, defense, things like that. And then I became the science editor. And then while I was doing that, I started writing books mostly about recent human evolution, based on all the new information flooding out of the human genome sequence.
What did you learn being a science writer about science itself?
Well, I think of it in sort of two parts. There's the pursuit of pure knowledge, and that's the substance of science, and it's mostly what gets into the newspapers. But science is also a career. It's a struggle for funding to beat out rivals, and this is a very important part of science that is less often seen. It was first brought to light in Jim Watson's double helix. But it happens all the time, and I think it has a powerful shaping influence on how science is done. So I feel probably more attention should be paid to that side of science by science journalists.
Do you think the public perception of how science works and the reality of how science might manifest itself ultimately with political influences, with financial influences, do they differ? Could the public be a little too trusting when they hear scientific reports or hear scientific opinions in the news?
Yes, I think so. I think they probably should be more skeptical. After all, the bread and butter of science journalists is reporting about the great things that scientists are doing. Here's another advance in treating cancer, and often it's not quite as big an advance as as is made out, because scientists and journalists have, excuse me, have a common interest in making things sound really great when they may just merely be good, or even not good. So that's one thing that has to be kept in mind. Another is that with the movement of our academic community as a whole to the left, this has politicized and, in my view, corrupted many normal venues of scientific discourse. So the scientific journals can no longer be trusted in the same way as we trusted them before, because they too have unfortunately failed to resist this politicization. And if you just look at any of the editorials, you can see how suffused they have become with sort of academic political issues and with the agenda of the far left, which is extremely short-sighted of their editors, it seems to me, because if their editorials are to the far left, how can you trust the scientific papers they publish? After all, the whole reputation of the journals and of scientists rests on being objective. So either you can be objective or you can be political. You cannot be both.
I've found something similar in reports on sociology and criminology, that there's influences on what is being reported, and perhaps in the scientific studies themselves. Don't scientists have ethical considerations when it comes to being, as you said, objective and reporting the objective truth?
Well, they do have those concerns, but they also have careers to pursue. So generally, there's great temptation to do whatever it takes to get a paper into a prestigious journal so as to help with your next grant. So if that means shading the truth, sort of bending a little so as to please the likely referees or the editors, especially if you believe they have sort of political opinions one way or the other, then that's what you'll do. I imagine if you're a climate scientist and you've decided that the climate is not warming, and you try getting that paper into Nature.
You have written a book. You've written a book on COVID or SARS-CoV-2 as well. What about this issue, SARS-CoV-2, piqued your interest?
Well, I should say I haven't written a book on the subject. There are many books, some of them very good. I've just written a series of articles. Well, the first one I wrote in May 21, which got a lot of attention, was based on an article I had read by a guy called Yuri Dagin, who's a biotech entrepreneur. And this just analyzed the SARS virus and sort of looked at every part of anatomy and sort of showed how you take it apart and put it together again. It was a very long paper, about 10,000 words, somewhat technical, but I just found this a revelation because it raised the possibility, though it did not endorse it, that the virus could have been engineered in a lab. So I sat back and waited for this remarkable page to be covered by the mainstream media, but no one said a word about it, which was extraordinary when you think of it. And here's the story of the decade, surely. And here's a serious article saying this is how the virus might have been engineered. Surely that's worth bringing to your readers' attention, but apparently not. So after a while, I started trying to write articles myself, covering the ground Yuri Dagin had opened up. And I found my articles were turned down every place I offered them. So eventually, in desperation, I thought, well, I'll self-publish this on medium.com, which, by the way, was where Dagin's article appeared. Write it out of my system, get back to the book I meant to be writing. So to my surprise, it got about 1 million page views on medium.com and the similar number in the Bulletin of Atomic Scientists, which reprinted it verbatim a few days later. And this, I think, sort of opened up the, according to some media analysts, it was this article that brought the issue back into media attention, the possibility that the virus had been manufactured in a lab, which previously no one had deemed worth even discussing. Now at last the discussion started.
Is it properly termed SARS-CoV-2, COVID? What term do you prefer?
Its formal name is SARS-CoV-2, but you can abbreviate it to SARS-2.
Okay. And this particular disease, I do recall when I first heard about it, it's in Wuhan, China, and in Wuhan, China is a laboratory studying viruses. I mean, is that correct?
Yes, China's principal laboratory for studying coronaviruses, which the family SARS-2 belongs to, is in the Wuhan Institute of Wuhan Institute of Virology.
Well, just that fact right there seems to stand out, I think, to even lay people or anyone that has an interest in it, that this very place happens to be a location where they have a facility studying viruses. I mean, wouldn't that simple fact cause curiosity that it could have come from that location?
You would think it gives a pretty good basis for at least raising the question of whether the virus might have escaped from the lab. And yet there was such a sustained and successful campaign to suppress the lab leak hypothesis that the mainstream media and most of the scientific community for months just completely dismissed this out of hand, believing instead the opposing theory that the virus leaped from some natural source to humans.
I could say, as an example, Frankenstein's monster looks and acts quite a bit different from a regular person from fiction. So I would think a virus that was manufactured would look and act quite a bit different than one that was natural. Is there something about SARS-CoV-2 that is very unusual just in the initial study of it that would make you think it is not a natural virus?
Yes, there are several features of the virus that make it look very much as if it's an artificial virus, one that has been synthesized in a lab. So the first thing is it has a small genetic element called the furin cleavage site that is responsible for its enormous infectivity and the ease with which it infects human cells. Now, this sort of leaps out to any virologist because none of the other virus members of SARS-2's family possess a furin cleavage site. So this is important because in nature, viruses can gain new genetic elements by swapping them with members of their own family. And that's how normally you would expect SARS-2 to have acquired such an element. But it cannot acquire any element that no member of its family possesses. And we now know of 870 members of the SARS-2 viral family. They're called sarbecoviruses. And not a single one other than SARS-2 has the furin cleavage site. So that is very anomalous and hard to explain in terms of a natural origin, very easy to explain in terms of a synthetic origin, and not least because we can see in the scientific literature that virologists often talk about inserting furin cleavage sites into their viruses to soup them up. The second feature, I'm not sure how technical to get here, is that if you look at the codons that specify the 4 amino acids in the furin cleavage site, these codons are human preference type codons, not bat preference codons. This is because each species has its own characteristic preference for the various codons you can use in the genetic code for specifying amino acids. So it's very, very indicative when you see a bat virus having human preference codons, and the easiest explanation is that if you were inserting the furin cleavage site, if you'd synthesized it in a lab, you would reach for a lab kit, and these lab kits, being mostly used for medical purpose, have human preference codons. So that is an easy explanation for where the human preference codons come from. Very hard to explain on the natural origin side. And the third thing, which I think is rather important, and that has only recently come to light, is that if you're going to synthesize one of these viruses, they're quite large viruses, 30,000 base pairs. So you want to break them down into sort of even-sized chunks. And if you were going to make SARS-2, you would probably you'd break it into 6 chunks and synthesize the DNA in the lab and then join them all together. And you'd use a characteristic tool, a restriction enzyme for doing that. So in 2022, 3 scientists predicted that if SARS-2 had been made by this usual technique, there should be the sort of fingerprints in its genetic structure showing how it was breakable down into 6 pieces. And moreover, the breakpoints, which in nature occur randomly, should be evenly spaced, because that's what a genetic engineer will do. So lo and behold, they found these 6 evenly spaced segments in SARS-2 and said, well, this looks like it was a synthetic virus to us. So this paper was poo-pooed by virologists who called it kindergarten molecular biology, and it didn't get nearly the attention it deserved until last month. Some documents retrieved by an organization called the US Right to Know under the Freedom of Information Act,
Mm-hmm.
found some of the planning, documents for a big grant that had been made to the grant application made to the Defense Department in 2018, and the authors of this grant included Shi Zhengli at the Wuhan Institute of Virology. So this grant said we're going to synthesize viruses in 6 sections,
Mm-hmm.
and we're going to use this particular restriction enzyme exactly as had been predicted
Mm-hmm.
by the authors of the 2022 paper. So this is a pretty strong indication
Mm-hmm.
that SARS-2 is a synthetic virus. Those are a long answer to your question, those are the 3 characteristic parts of the SARS-2 genetic structure that point to a synthetic origin.
So an earlier paper stated that if this is manufactured, you will have the virus into these 6 different parts. There's something you could actually see. And now we have a recent document saying there was research to do that very same thing.
Right. And now this document is significant because it relates to something called the defuse proposal, which is the most important sort of documentary evidence we have bearing on the origin of SARS-2. And this was a grant proposal made in 2018, headed by a guy called Peter Daszak of the EcoHealth Alliance in New York.
Mm-hmm.
And he had 2 teams of scientists, one by Ralph Barrick in North Carolina, and one by Zhengling Shi in Wuhan.
Mm-hmm.
And they, in this proposal that was leaked a couple of years ago, they said they were going to manipulate SARS-1 type viruses. SARS-1 is a bat virus that caused an earlier epidemic, a brief one, a smallish one in 2002.
Mm-hmm.
So they were going to work with viruses related to SARS type 1. They were going to insert furin cleavage sites into them.
Mm-hmm.
And that meant that depending on their starting virus, they could have produced SARS-2 by this method.
Mm-hmm.
Now, this grant was turned down by DARPA and sort of sunk from view somewhat until the new batch of documents released last month, which were all the planning documents
Mm-hmm.
for the defuse proposal. And it's from these planning documents that we can see how very closely their recipe for making viruses was to what we know from the structure of SARS-2. Now, if I can add just one more thing,
Mm-hmm.
when DARPA turned down that grant in 2018, possibly because they thought it was far too dangerous, that doesn't mean the experiments weren't done, because Barrick and Shi, although they were collaborating in this grant, they were also scientific rivals.
Mm-hmm.
So it's very likely that Zhengling Shi, once she had seen that Barrick was halted because there was no money there from DARPA,
Mm-hmm.
she realized that if she could get money from the Chinese government, she could recoup all her investment in planning for this grant and race ahead of our rival Barrick.
Mm-hmm.
Now, this fits very well with what we know actually happened, because if she'd started making these viruses in 2018, she could have finished making them in 2019,
Mm-hmm.
which would explain an otherwise totally unexplained fact, which is the timing of the epidemic. And this SARS-2 epidemic could have occurred any time in history if it really came from bats. But no, it occurred in this very small window of time, probably August, September of 2019. And this matches precisely the timing of she carrying on with the defuse experiment with her own funds. And of course, it also matches the origin, the place of origin of the virus.
it is surprising, it was to me anyway, I think it is to others, that I didn't know before SARS-2 that we now are in an era when scientists can change viruses significantly and can actually make them infective to people when they weren't previously. And I believe that's called gain of function research. Can you talk about that a little bit?
It is surprising, but now gain of function research has been going on for about 10 years, and it's been sponsored by the National Institutes of Health, and particularly by the National Institute of Allergy and Infectious Diseases, which is one of the component institutes of NIH, which was headed by Anthony Fauci. And he and Francis Collins, the head of NIH, jointly wrote a letter, I think in 2014, in the Washington Post, saying, yeah, this new research we can do is hazardous, but we think the benefits make it well worthwhile. And they proceeded to fund this kind of research. Now, there was quite vigorous opposition from many scientists who said, this is crazy, this is dangerous, the benefits are not conceivably worth the enormous risks. But Fauci and Collins paid no attention to these objections. I think this was a very serious mistake, because they ignored a very good precedent that the scientists had set up much earlier when they first learned how to manipulate genes. They held a big conference. This was in 1975. It's called the Asilomar Conference. They said, here we have this new ability to swap genes from one organism to another. It could be dangerous. So let's start out by setting very high safety standards. And if the research turns out to be not so dangerous as we fear, then we can progressively reduce the strictness of the lab security. And that's just what happened. So this was a very good role model for how to handle really dangerous research. And what Collins and Fauci should have done when gain of function research, enhancing viruses, first became possible, they should have held a worldwide scientific conference of the very best experts from many disciplines, not just virology. And they should have figured out a safe way to approach this research. And if they had done that, then when this epidemic occurred, they could, and if it was caused by the escape of a virus, they could have said, we were acting on the best advice that we could get hold of. But they can't say that because they ignored all this outside, the outside critics. They went ahead and in a bureaucratic way, they sort of stifled most of the criticism and objections. And as part of this program, they funded this wildly dangerous research at the Wuhan Institute of Virology, which should never have been funded whether a virus escaped or not. And that is why we find ourselves in a position where this dangerous research is still going on, and no one has come forward and said, look, one bad mistake is enough. Let's reassess, because the scientific community has not yet come to grips with the fact that this virus may well have escaped from Wuhan. So they are not yet being forced to face the serious consequences. The gain of function is something that should not be done lightly.
Yes, I feel the need to just go ahead and ask, does anyone know what's going on in the Wuhan laboratory currently? Is there any checks, or anyone got a chance to go in there and see? Do we know what's happening in Wuhan now?
No, we don't. The WHO sent a sort of big scientific commission to look into the origin of COVID. They were completely stonewalled by the Chinese. They were not allowed to see anything. I don't know if they were even allowed to set foot in the building, but if they were, they weren't allowed to see anything that they wanted to see. The critical bit of information we need is what viruses were being worked on at the Wuhan Institute, because then we could see whether the precursors, the likely precursors or parent of SARS-2 were in that collection. But the Chinese have refused to show the lab notebooks or the viruses being worked on, and they closed down in I think September 2019 a very important database of SARS-type viruses that they had maintained and was open hitherto. So the Chinese have been totally uncooperative.
So you have lack of cooperation from the Chinese, from Wuhan. I had read a report of a modification of ventilation systems in the Wuhan laboratory around 2019, possibly suggesting either they felt that the ventilation systems were unsafe or that something had happened that they decided they needed to make these modifications. Have you heard similar reports?
Yes, I have. That's very interesting sort of circumstantial evidence. I mean, it doesn't bring you nearer to sort of proving where the virus came from, but there are lots of pieces of evidence like that that sort of combine to make a quite impressive overall picture of a sort of large-scale government cover-up.
Does it appear to you that there is not just disagreement on whether it was a lab leak or a natural origin, but there is active resistance in the scientific community, perhaps elsewhere, to the lab leak hypothesis? And if that's the case, why would that be?
Well, I think it's fairly clear there was a campaign directed by Fauci, by Collins, and by Jeremy Farrar in London, who's head of a big medical research organization, to suppress the notion that the virus had escaped from the lab. And the reason I say that is that the first group of scientists who looked into the origin, headed by a guy called Christian Anderson, who was a grantee of Fauci, that little group sent an electrifying email to Fauci on the evening of January 30, 2020, saying, essentially, we think this virus is manufactured. It doesn't accord with, we can't explain it in terms of evolution. In other words, we don't think this virus has a natural origin. So I think they thought Fauci might be very pleased with them for having found out the origin of this terrible epidemic. They learned Fauci wasn't at all pleased to have this answer, and he arranged a conference the very next day at which several scientists who were very much in favor of gain of function attended, as well as Fauci, Collins, and Farrar, and along with Christian Anderson and his team, who decided this virus did not have a natural origin. And at this conference, although we don't have a transcript, it seems that the role of the pro-gain of function scientists was to sort of poke holes in the Anderson team's assumption. And at the same time, it seems it was made clear to Anderson that he'd got the wrong answer. Because within 2 days, without access to any new scientific information that we know of, he completely changed his tune and started saying, well, lab origin is a conspiracy theory, and here's all the reasons why it's wrong. So out of this teleconference grew 2 papers in the scientific literature, which were enormously influential. One was in the Lancet, and the other was in Nature Medicine.
Mm-hmm.
And it was these 2 papers that essentially buried the lab leak hypothesis. And it seems, this was clearly by design.
Mm-hmm.
It's clear that Farrar, for example, sort of midwifed the paper in Nature Medicine and sort of called all the shots on it. And Anderson and his colleagues sort of, wrote it until everyone was happy that they had not done the best job they could to knock down the lab leak hypothesis. And they were enormously successful for at least a year. So why did this happen? Okay, this is more a matter of speculation. I think the different players may have had different motives. I think for Farrar, he had very close connections with Chinese scientists, Chinese health officials, and
Mm-hmm.
I think he wanted to assure his, do a favor for his friends. For Fauci realized very early on that he had funded the research at Wuhan. So imagine his dismay when Anderson tells him that he thinks the virus has escaped from the lab at Wuhan. And Fauci knows that, well, he has funded this research, not directly, but via
Mm-hmm.
the EcoHealth Alliance in New York, which was his sort of cutout. So Fauci had every reason to try and squelch the idea of lab leak. For the virology community as a whole, virologists, I think, were very afraid that there would be enormous public backlash against them for having generated this virus. And even if it was the people at Wuhan were the main ones responsible, nonetheless, the Chinese scientists were sort of working in a framework, in a research framework established by American and European scientists. So in essence, the virologists were all in it together. And I think they had every motive to try and forestall this sort of serious threat to their funding. So that, I think, explains their motives. And then if you move to the media, somehow or other, and I think Fauci probably had a lot to do with this, that I can't prove it, the issue from very early on became highly polarized.
Yeah.
So if you're on the left of the political spectrum, you favored natural origin. If you're on the right, well, you didn't really care very much, but at least you were open to the idea of a lab leak. And because of that polarization, the mainstream press, which is largely to the left, has never seriously reported the evidence in favor of lab leak. Every time there's counter evidence in favor of natural origin, they put that on the front page. So the public, by and large, is very ill-informed on this issue, in my view.
Do you know of any political leaders, heads of state, who have spoken openly and supported the lab leak hypothesis, or at least its investigation?
The only political leader I've heard of who is open to the idea of lab leak is Michael Gove, who's a cabinet minister in Britain, who's said words to the effect of this seems probable, or this should be looked into.
What are your thoughts? I do feel the need to ask this on the possibilities some have raised that SARS-2 was not simply an accidental leak, but was an intentional release. Is there any evidence to that effect?
Well, this is a very interesting idea, especially in the early days, because it's clear that these bat viruses were first found in a cave in southern China, which is hundreds of miles away from Wuhan, where the epidemic broke out. So what is the link between these two places? Well, one obvious link is we know the Chinese, excuse me, we know the Chinese researchers in Wuhan made frequent trips to these Yunnan caves to collect bat viruses, and they brought them back to their lab. So that could be the link. Some people suggested these were natural viruses brought back by researchers, which then escaped. So that's an interesting idea. However, when you look at the many signs which we just discussed that point to the synthetic origin of the virus, then that sort of rules out that particular explanation. The viruses were brought back from Yunnan and Laos and other places because the Chinese had a vigorous virus collection program, and they were taken to Wuhan, but they were then experimented on and changed and altered, and it's one of these altered viruses that has escaped.
They were able to capture a real virus. They completed their alteration of it for these scientific reasons, and then more than likely, due to some kind of negligence, it was able to escape the laboratory, and now this altered virus is out in the public. It's in the public among
Well, that's what seems to
yeah
that's what seems to have happened. We don't know that for sure, but it looks more and more like that is the cause of the epidemic. Now, one reason is that the Chinese were working with this virus in very low level safety conditions. Xi Zhengli did most of our virus work in a type of lab called BSL-2. There are 4 levels of safety for these biological labs, and BSL-2 is very low level. You basically sort of slap a warning sign on the door and don't suck up liquids with a pipette. That's more or less it. None of this sort of spacesuit negative pressure type environment you see in a BSL-4 lab, which is the highest. Now, scientists don't like to work in BSL-4. It's very encumbered, it's very costly, it's cumbersome, everything takes twice as long. So she did all her work in BSL-2. Now that contrasts, although that was permitted by international regulations, it stands in sharp contrast to the practice in the US, where the chief coronavirologist, Ralph Baric, believed these viruses were seriously dangerous, and he did all his work in BSL-3 type labs. So the Chinese were using the wrong safety conditions to work with this virus, and it should be no surprise that the virus did indeed escape.
I would think something like this would be at the highest level of safety all the time, as if it was as dangerous as nuclear material or something to that effect. If it can kill millions of people, I would think it would be at the highest safety precautions.
Well, that's right. I mean, in the first place, you should never make such a virus. And in the second place, you should, at the very least, do it in BSL-3, as Baric has done, I think, without any accident so far, and if not, BSL-4.
If we had access to all evidence, the Wuhan laboratory, everything from these different parts of the world, the United States, China, wherever, without restriction, what tests or analyses do you think should be conducted to determine the origin of COVID-2?
Well, unfortunately, there's only one test to prove it, and that is to find the parent viruses from which it was derived.
Mm-hmm.
So the diffuse proposal, assuming that she did go ahead with it unilaterally, the diffuse proposal says they're going to take a batch of SARS-1 type viruses, and they're going to see which are best at infecting human cells. And then they're going to make a consensus sequence virus. So virologists often do this. It's a sort of cunning trick. You find out what are the best viruses that nature has produced, and you sort of combine the top strengths of all the best viruses to give you a consensus sequence. So the diffuse proposal says they're going to get this consensus sequence. They're going to make 10 or 15 viruses, as I recall, and they're going to synthesize them with this sort of 6-part method. So the only way you can prove that that is how we got to SARS-2 is to find the parent viruses that she would have used to make her consensus sequence, or to find one of the consensus sequence viruses. That is the only sure proof. And since I don't think we're going to get that in the foreseeable future, you really have to ask if we really need that final missing piece. Do we not have enough evidence already from the diffuse proposal, from the actual structure of the virus, to make a pretty strong case that SARS-2 is a synthetic virus? If so, we don't need that last missing piece.
If it became more widely accepted that the lab leak theory was accurate, what do you think would happen in terms of public policy? What changes would result from this information?
Well, that's a very interesting question. I can only speculate. I think what should happen at a minimum is there should be a thorough review of gain of function experiments, that the power to fund them should be overseen not by the NIH, which has a conflict of interest. It should be overseen by some independent overseer who will say, we know what experiments are worth the risk of doing and what safety conditions should apply. So that's just a very minimum. And this should be done internationally. So everyone should have their same, hopefully uniform system of independent regulation of this extremely dangerous research. Then you have to ask about the role of the virology community, which not only has set up the framework for this gain of function research to go ahead essentially unregulated, but has then sort of circled the wagons and done every possible thing they can to deceive the public as to the possible origin of this virus. I think they are very much to blame. I think the public likes professional bodies to regulate themselves if they can do so responsibly. I think virologists have clearly failed to take a responsible attitude to this research. And someone needs to bring the hammer down on them and say, you guys have not behaved like adults. We need to have close supervision of what you do. We don't care if it retards your precious research a bit. Most of what you were doing with gain of function was pretty useless anyway. The benefits you promised have not arrived. We're going to regulate this carefully, or maybe stop it altogether. And your funding is going to go down. Why should we fund you to put us all at risk? Now, I think that kind of backlash, which would be completely justified, may also apply more widely to the scientific community as a whole. Now, I think this would be unfair because many other professions which are sort of developing more dangerous tools by the minute because of the enormous power of molecular biology. Many other groups of scientists, if you look carefully at what they're doing, are behaving very responsibly. For example, there's something called a gene drive, which has enormous capacity to eliminate disease vectors, but also enormous risks if the drive slips out of your control. So the people doing gene drives have been very open. You can sort of read their discussions, their attempts to bring this to public attention, to get public permission for everything. These scientists are behaving very responsibly. So it would be a pity if the virologists' bad behavior should impede research across the board. But that is a possibility. It all depends on how annoyed or outraged the public is at a small group of irresponsible scientists who allowed the death of 7 million people or more around the world.
It sounds like the scientific community's reputation and those of the medical community and governments have been seriously harmed by this entire crisis.
Well, I think they will be seriously harmed on the origin question. And of course, there's now all kind of second guessing on the sort of vaccine and masking issues. I'm not an expert on those. I feel the vaccine authorities have a very difficult position because, and we should cut them some slack. And the reason is that on the one hand, you need to present a confident front to the public saying, yes, you should be vaccinated. On the other hand, you usually don't have the absolute certainty you would like because most vaccines have side effects down the road. You hope very minor side effects. They don't negate the enormous benefit the vaccines have, but it's very hard to sort of say to the public, this vaccine is completely safe, because really, although vaccines are much, much safer now than they used to be, you can't really guarantee any vaccine is totally safe. So it's very hard. It's a hard line for a public health authority to walk. So it's very sort of distressing when Fauci tells us, for example, that this sort of separation rule, everyone should keep 6 feet apart, that this rule had no scientific basis that he could think of. I mean, you shouldn't behave like that if you're trying to keep the confidence of the public. You need to have, sort of maintain a strong relationship with the data you have available and the best advice you're trying to give on the basis of that. So I think this whole process now of second guessing what was happening, although the criticism I think is in many ways too virulent, nonetheless it's an important exercise in self-examination, and I hope it leads to sort of better vaccine public health policies in the future.
Excellent. So what are your hopes for the future at this point? That covers it perhaps a little bit, but for you, what are your hopes for the future on this whole issue?
Well, the origin issue, which is the thing I've studied most carefully, I do think we're sort of pretty near certainty. We don't have the absolute proof that will be provided by knowing the parent viruses, but we've got something which is pretty good and pretty close to that. And I hope that, especially with this sort of new batch of documents that was released last month. So I hope as awareness of that spreads, people will begin to realize that lab leak is the most probable explanation for this epidemic, and that they will then proceed to take some of the steps we've just discussed.
Okay. Nicholas, I really appreciate you taking your time to talk to us about this very important issue. I learned some things about it. I learn new things all the time about SARS-2. I'm not really sure where it's headed in the future, but this is pretty interesting information, especially these recent developments in the last month. So I appreciate your time, and if we could have a chance, maybe there'll be future developments and we could interview you in the future.
Sure, I'd enjoy doing that. Thanks for your interest.
Okay. Thank you, Nicholas. Have a good night.
Same to you, Timothy.